Today's lesson is bacterial behavior, eating, growing, reproducing, etc. All the stuff that bacteria do. Well, one thing that they do is they occupy all parts of the food chain. For starters, they are consumers, which means that they consume other organisms in order to survive. If the organism you are consuming is dead, then you are called a saprophyte. You are a saprophytic organism if you eat dead stuff. And if you eat dead stuff, you're probably a decomposer. That is, if you're a bacteria. And your job in the ecology is to change dead organic matter into raw materials that other things like producers can use. Although, if you're not eating something that's dead, you're eating something that is alive, you're probably living in it or on it. And in that case, that bacterium is called a parasite. That is an organism that feeds on a living host. So anytime you've ever had an infection, that's bacteria living in you, benefiting off of all the nutrition that your nutritious body has to offer it when it eats it, then you have a bacterial parasite. This is not a happy subject. Let's move on. Producers, that's much nicer. So if you are a bacterium or any organism that produces your own food by harnessing energy from sunlight, well, that's photosynthesis. And if you're doing it by harnessing energy from a chemical source, that is called chemosynthesis. So bacteria can be consumers or producers. They can also live in all kinds of conditions. With oxygen, if you need oxygen in order to live, you are called an aerobic organism. But if you do not need oxygen to live, you are an anaerobic organism. In fact, oxygen is poisonous to some organisms, and that's a way that you can get rid of them. You might know the word aerobic, like from aerobic exercise. That would be where your heart rate goes up, your breathing rate goes up, so you're using up more oxygen. See how that works? Yeah. So reproduction in bacteria. All that you have to do here is have enough material to share between two cells and then just split in half. That means that you need to have enough cytoplasm and enough ribosomes, but it also means that you need to copy your DNA. Have one copy of DNA on both sides of your cell and then just pinch and separate. That process is called binary fission, and it's, well, have a look for yourself.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Yeah, so this is why you want to brush your teeth at night, right? Before you go to bed, I mean. So population initially grows exponentially. What that means is really fast. But once there's a whole bunch of bacteria, then the population reaches a ceiling where the resources don't support more growth. Could be because the food source isn't enough to support more bacteria, or it could be that there are so many bacteria that they're competing with each other for resources. You end up with this steady state. It levels off. And so if this is exponential, the shape that this makes, where it rises exponentially and then levels off, is called logistic growth. That's a fancy math term you don't need to know. All you need to know is bacteria grow real fast, but they can't grow forever. So genetic recombination in bacteria is how you end up with different kinds of bacteria. If bacteria can only ever clone themselves, then why aren't all bacteria clones? Here is a good explanation for why. First of all, you've got conjugation. That would be an exchange of DNA between two bacteria. Bacteria can actually share DNA with each other, and that results in a modified, you almost might say mutant version of the bacteria, because your DNA kind of defines who and what you are. If your DNA changes, you change. Transformation. That would be where a bacterium dies, its cell breaks apart, its DNA is just floating around. I mean, what's going to make it decompose? Bacteria? See? Anyway, so the DNA is floating around. Another bacteria comes and eats it up. It's got this bacteria inside it. It's got this DNA inside of it now. New DNA, slightly changed bacterium. And also transduction. That would be infection by a virus that results in DNA being transferred from one bacterium to another. Believe it or not, bacteria can get sick, so to speak. There is a kind of virus that only targets bacteria. It's called a bacteriophage. And they can transfer DNA from one bacterium to another. Absolutely crazy stuff. So when you've got bacteria that are in good conditions, you've got that kind of exponential growth that we were talking about. But if you're trying to kill bacteria or it's just not a good place for them to live, they can still manage to survive with an endospore. That's where the DNA and other essential parts of a bacterium are coated with several hard layers. And these protective shells are in response to extreme conditions. They can protect it against extreme heat and cold, against a lack of water, even against toxic chemicals. So, of course, I wouldn't expect very many bacteria to survive this way, but this is a kind of last resort that bacteria have when things are getting kind of lethal for them, that they can just protect their DNA and their vital parts in hard layers and make it through. Once the conditions become favorable again, then the bacteria can just reemerge from this endospore and begin being all bacteria-y again. Okay, last for this lesson is bacterial colonies. Individual bacteria have their own behavior, but one thing that single cells can do is live in groups. We call those colonies. And the names for these colonies are based on what kind of bacteria are in them and also how many bacteria are in them. So if you are spherical bacteria, if there's just two in a colony, it's called Diplococcus, two bacteria. Remember diplo, like for two. If there are four, we call that Tetracoccus, four spherical bacteria in a colony. Streptococcus would be a long string of bacteria, so we go from 2 to 4 to just many. And if the long string is kind of branching, kind of like a web, it's called Staphylococcus. Maybe there's a familiar sound in there. Ever heard of a strep or a staph infection? Those are related words, aren't they? And if you're a rod-shaped bacteria, you might remember that was bacillum. If there's two, we call that Diplobacillus. These are so much fun to pronounce, you guys. Streptobacillus, a long string of bacteria, right? Strepto, it's the same thing above and below. And there isn't any names that I could find for colonies of spiral bacteria. Maybe they don't make any. Anyway, that is bacterial behavior, at least for today. Have a good day and avoid those bacteria.